Thank you, Yves.
So the topic today is the basics of radiation biology for radiation processing. This is a topic that has not very often been covered by the IIA. So the IIA dealing with radiation processing, I took, of course, a certain angle to it. So what will we discuss today? I'm going to start with a brief reminder of the key concepts, very briefly, things that you have heard before, but that are absolutely essential to understand the rest of the presentation and to understand what radiation processing is about. Then we'll talk about radiation biology, and it will revolve very much around the DNA, of course, the molecule of life, sensitivity to radiation of various living organisms. When you handle radioactive sources or machines producing ionizing radiation, of course, radiation safety is essential. So I'll say a few words about this. Then we'll see what I've called radiation biology in action, having a look at different applications of radiation biology with, let's say, different effects on different living organisms that are used as a research tool or in practice, not so necessarily on a commercial scale, to have the beneficial effects of radiation. So to start with the basic, I'm going to use a slide that I used during the first webinar, where we'll be discussing ionizing radiation. And they are called ionizing because they induce ionizations in matter. So we know that an ion is created when an electron, for example, is ejected out of its orbit. So this can be achieved with photons from X-rays or from gamma rays, or from electrons from accelerators. At that point, the electron that is ejected will go very much like the balls on a pool table, kick other electrons and interact with matter, so forming ions, and these ions will give birth to radicals. Radicals are very reactive species, and they are responsible for the chemical effects rearrangement that will later occur. I think you know by now, if you attended the previous webinars or if you've been in this business, that irradiation is essentially a transfer of energy by which the energy emitted by a source is deposited in a target. The key parameter is the dose, and the dose is simply the quantity of energy absorbed per mass unit. The joule, the gray, is the unit, and when 1 joule has been absorbed on 1 kilo of matter, you have delivered a dose of 1 gray. So fundamental is that, speaking of effects of radiation, there cannot be general statement really, because it all depends on how large was the dose. So you have very, very low dose that will not have effects. And you can have, for example, when you treat food, when the dose is high, or I would say too high, in the same way as if you cook a product too much, you will have undesirable effect. So anyway, very broadly, the chemical effects and the biological effects will increase as the dose increases, well, in different manners, as you can see in this very simple graph. So radiation biology is the branch of biology concerned with the effects of radiation on living organisms. You see on this slide that there are many effects of radiation on very different in very different areas. You have cosmic rays that are ionizing radiation that bombard us. We know, of course, of cancer therapy. We know there are controversies about the effect from effluents from radioactive plants. We deal with also radiation when we have our luggage scanned in the airports. In the airport. So many different aspects actually of radiation biology. And radiation biology does not only deal with irradiation, but also with contamination, which can be simply described as the presence of radioactive substance inside or on the surface of a living organism. So this is not what we are talking about. Though this is also interesting, if you call about cancer therapy, for example, you can implant radioactive sources inside a body and have beneficial effects from the radiation that they will emit locally. So here we're going to talk about exposure to irradiation. Even if you do not have a PhD in biology, you probably know that the cell is the basic unit of all living organisms, of all living things. I wrote because, well, when I was a student, it was simple. You had plants and animals. Now it's much more complicated, and you have things like blobs, for example, which are strange. They're strange living organisms. So here you have the representation of a plant cell and an animal cell. They differ, but for example, a plant cell is characterized by a wall that forms the membrane. But all cells will have a nucleus. This nucleus will contain chromosomes, and the chromosomes are made of DNA, which is the program of the cell, giving the instruction on what the cell should do. So the main damage of ionizing radiation will be on DNA. We will see that this changing can lead to the death of the cells through different processes. So when you talk about effect on living organisms, actually, it's not biology at the base. At the base, it's chemistry through the effect on DNA and other molecules also. And I cannot go into this detail because DNA is indeed the main effect, but proteins, for example, those linked to membranes, etc., can also be affected by radiation. I don't know how to go to the next slide. Oh, okay, I got it. So DNA, how is the DNA is affected? Well, when you bombard a cell with radiation, there are two routes. One possibility is the direct route by which the radiation will directly hit, let's say, the DNA molecule. And several modifications will ensue. However, this is not the main route. The main route is indirect. All living organisms contain water, a lot of water, and the chances are that the radiation is going to hit a molecule of water before it hits a molecule of DNA. And it happens that the releases of water, so the effects of radiation on water, will lead to a number of radicals, so again, very reactive species. And it is these radicals that have the most effect on the DNA molecule. So again, the main effects come through indirect effects of radiation. So now what are the damages? What you can have is, for example, these two the two strands of DNA can be broken, both broken, or only one can be broken. And you know that these two strands are linked by weak bonds, hydrogen liaisons at some point, and these bonds can be made covalent by chemical changes, and so it will like tie the two strands together or fragments of the strands together. At a dose that would be considered low, certainly in radiation processing, because radiation processing is generally in kilograms. But if you look at cancer therapy, we are much closer to these low doses of a couple of grays or even less. So you will have roughly 1,000 more damage to the bases, about 1,000 breaks of a single strand, and 40 double breaks of double strands. And these are the most damaging ones. They totally break the molecule and will make, for example, the replication process more difficult, if not impossible, for the particular molecule that was hit. So you will see a number of consequences, as you see on the picture at the bottom right, with fragments, the chromosome being broken. Excuse me. Now, we know that nature is it's so well organized that actually cells are able to repair damages, and DNA in particular can be repaired thanks to a number of enzymes that cut, paste, etc., so that can repair different types of lesions, of damage to the DNA. This, of course, again is going to be in view of the doses that are used. In radiation processing, for most applications, the dose is so high that the damages are just too many for the cell and this repair mechanism to cope with. So the DNA will be definitely damaged. When the DNA is not repaired or incorrectly repaired, that will lead to modified chromosomes and aberrant chromosomes. So you have a number of possibility of moving the different pieces around, etc., that will create damage to the cell and eventually lead to its inability to reproduce itself or to its death. It can be, to put things simply, it can be said that the more complex a living organism is, the more sensitive it will be to radiation. That has to do very much with the size of the DNA. You know that humans, for example, have 23 pairs of chromosomes, so that's a lot of DNA in each cell. At the other end of the spectrum, you will have organisms like prions, for example, or virus, or even bacteria that have a lot less DNA. So it's as if the human cell offers a very big target, DNA target, to radiation, when more simple organisms have only a small, or represent only a small target, so less chances of being hit. So this is how you have a scale from the most sensitive organisms, the humans, and to the most resistant organisms, like, for example, the prions. In 1906, two French radiobiologists came to a couple of principles that are still valid to this day regarding the radiation sensitivity of cells to ionizing radiation. Actually, the use of radiation for cancer therapy in particular came very soon after the discovery of radioactivity, and for imaging and for radiation therapy came very soon after the discovery of radioactivity and X-rays by Röntgen. So already in the early 1900s, they were starting to understand the sensitivity to ionizing radiation. So these gentlemen, Bergoni and Tribondeau, stated that the cells are radio sensitive when they have a high division rate. For example, these cells that will be in the middle of mitose when they are hit by radiation. When they have a long dividing future, meaning that these cells are designed to give birth to many new cells, and this is the case, for example, of spermagonia. So these entities of namely the spermatozoids and the ovules in the body, they are meant to divide many times to give birth to new beings. So this will be very sensitive to radiation. It's also the case of hematopoietic cells, so those that will form the different components of the blood that you find, for example, in the bone marrow. And finally, cells will be all the more sensitive that they are unspecialized. So these cells in particular, the spermagonia, will be able to turn in any kind of cell within the embryo, for example, and these will be very sensitive, unlike the cells that you will find in bones or collagen, for example, which are already very specialized and not meant to transform into another type of cells, and already having a very specific structure and function. A very important criteria regarding sensitivity to radiation is the linear energy transfer. So I was saying in the beginning, we talk transfer of energy. So the energy is deposited along the path of the radiation through the living organism, and it can deposit these the energy, as you see in the scheme below, at, let's say, in different points in close proximity, or in a more spread out manner in the cell. And then we introduce the concept of linear energy transfer, which is the average energy that is deposited per unit of length. So the unit that will be used will be kilo of electron volts that you could convert to joules, per micrometer. So a dimension that is commensurate to the size of the cell. And the more energy that the radiation will deposit on a very short distance will mean a higher linear energy transfer, and the more damage to to the living organisms. So some radiation have a rather low linear energy transfer. That's the case of gamma and X-rays, you know, because matter is kind of transparent to them. So the deposition of energy will be very spread out. But if you look at accelerated electrons, alpha particles, they are not very penetrating and they will get rid of their energy over a very short distance. And there is a particular case of neutrons that have also a very high linear energy transfer. So if you look, for example, in this graph at the dose delivered to mammalian cells, you see that comparing two types of radiation, one with a low energy, with low LET, and one with high LET, you see that for the low LET there is a shoulder, which means that there is still some capacity for the DNA to repair some of the damage. When this does not happen with high LET, where no or minimum repair is taking place. So in radiation biology, one important concept is dose equivalent. If you're a physician, you use grays, and it's simple, it's joules per kilogram. But it cannot be such with when you talk of biological tissues, because depending on the radiation, they will have different sensitivities. So the quantity that is used to characterize the energy has to be corrected by something that is called the quality factor. Now, as I made a note and I did not read it at the beginning, I have to simplify different notions. So, for example, the quality factor Q is a simplification. There are other weighting factors, as they are called, that can that come into play. But this is valid. You know, the dose equivalent is the product of the physical dose expressed in gray, multiplied by a quality factor that is usually named Q, that is actually related to what I said before, to the LET. So different radiation will lead to different quality factors. So you see the quality factor here for photons and energy in the small table is 1, also for electrons, but much more for neutrons, for example, or alpha particles, which unfortunately we do not come across very often in our daily life. So you have here just two values, the unit being the sievert, and I'm sorry because the unit is the sievert. So actually when the quality factor is 1, it's going to be the same number as the number of grays. So for humans, you see that the doses that are lethal are very, very low. So less than 5 sievert will be enough. It gives you 50% size risk or chances, if you want to call it like this, to die within 30 days. If you have received an irradiation of your whole body over a short duration. This would definitely be the case if someone were exposed to a gamma source. You know, in many industrial facilities, gamma sources contain millions of curies, and the dose rates are very high. So anyone coming near the source in irradiation position would be killed very rapidly, would die very rapidly after the exposure. And then if you increase the dose to 8 sieverts, well, your chances of dying would come up to 99%. So radiation is potentially dangerous. We'll come back on this right away, actually, talking about radiation safety. It's very important, I think, to mention radiation safety in this webinar, because of course it's a key aspect, though we do not often speak of it at the AAIA. Why? Because in irradiation facilities, especially the industrial ones, I would say, safety is built in by design. Many lessons were learned from experience. It's not that people were irradiated in irradiation facilities, you know, when the business started, no, but they became better and better, and actually the chances of having an accidental exposure are very low. But anyone working in this field must be aware of the hazards of ionizing radiation, and actually it is mandatory in irradiation companies to have a training on them. So the basic principle would be to minimize exposure time, because the shorter the time, the less the dose. Maximize the distance to the source. Generally, the dose will go down conversely proportional to the square of the distance, but these are not aspects that are the principles that are used in a radiation facility, because you have no exposure at all, and it's no question of going near the source, but not too near. So what is used in radiation facilities is actually shielding. So in a gamma facility, accelerators also, you have a bunker around the source, and when the source is working, you cannot go into it. So shielding would be the main way, with a number of redundant systems to prevent any exposure. It's important to note also, you know, that and public does not necessarily understand that people working in irradiation facility do not have any particular medical surveillance, because in a properly designed facility, nowhere the dose rate will be above 2.5 microsievert per hour, meaning that there is no, in most countries, regulatory obligation to have a special medical surveillance. So the personnel is, let's say, normal people, and in the category public. Now, about the safety of radiation, I would recommend this document published by the IAEA, where you can know everything about the safety of irradiation facilities, in particular, the design principle that make them safe. Irradiation, commercial irradiation, has been in existence for 70 years now. Yes, there were a few accidents, but really very, very few. And so this industry is an excellent record. And when there were accidents, and probably I would say, if you consider exposure of people, less than 10, and every time it's not the facilities that were the problem, it's the people operating it. And the accidents happen because they didn't follow the safety operating procedures, or they thought that they were smarter than the system, and they bypassed some safety system, and then when accident happens. Anyway, Safety, it's a never-ending story, and you have to work constantly on this, and by introducing what is called a safety culture in the organization, using irradiation commercially, but also in a research institute. So this has many aspects. It's a philosophy, it's the involvement of the management in particular to implement the safety. Now, something that I call radiation biology in action, the application of radiation biology. As we know, there are many potential applications. They will be detailed in the following webinars. And those that I want to discuss here are actually different effects of radiation on living systems. There are put at work to get beneficial effects. So we're going to talk about plant mutation breeding. So you see on the right top of flowers that is the result of mutation breeding. It was not commercialized, but you can get interesting things. We'll talk about the sterile insect technique, phytosanitary irradiation, and finally, the classic application of irradiation, which is pasteurization and sterilization. So plant mutation breeding is what? In nature, you have naturally occurring, if I have fields in front of me, fields of wheat right now as I speak to you, and there will be in this field spontaneous mutations to cosmic rays, etc. So mutation happens, and irradiation can be used as a tool to increase the probability of a mutation occurring. If you look at this at this little picture on top right, what you see is that mutation is about changing the sequence of the different bases, you see, on a gene. It has nothing to do with genetically modified. There is no foreign material introduced into the cell. It's only through the molecular effect of radiation on the DNA that, that you will obtain new varieties of plants in particular that will have new properties, interesting properties. We hear climatic change, you know, so plants that will resist to drought, for example, or sweeter fruit, or fruit that will mature earlier, new colors for flowers, etc., etc. So there is at the moment commercially available more than 1,000 varieties of plants in the world that have been improved really through the use of mutation breeding by irradiation. The IAEA is a database of this, and where there are about 5,000 different species, but the commercial ones are about 1,000. Now, increasingly, the accelerated particles are used rather than gamma or X-ray in the past, because let's say it's a more convenient tool to control, so to speak, what you're doing, because you don't really control, you go fishing. A number of mutations will be lethal or will not real mutation, because it will stop shortly, or will lead to aberration, making the plant non-viable. And when you want to increase mutation, you might think, oh, I'm going to increase the dose, but you know, the samples, remember that the samples must survive, not like for sterilization or pasteurization. You need the living organism must stay alive, of course, to produce new generations with new properties. So when we're talking food irradiation, for example, do not talk about mutations in the microorganisms that that the food may contain, because it's very different doses, and in particular, it's very different dose rates. The dose rates for mutation breeding are generally very low. It's chronic exposure very often, so it's the dose is delivered in fraction over a long period of time to make the mutation appear. So it's do not mix mutation breeding with sterilization, for example. Mutation breeding, interestingly, can also be done in space. You know, there were experiments taking different seeds in rockets and bring them back on Earth, and to see if there would be interesting mutations. Also interesting because in outer space there is a lot of radiation around, so mutations appear at a higher rate than on Earth. And then another technique that is based on a different effect of radiation on living organisms is the sterile insect technique. And this technique, I would say, is very, I'm an agronomist originally, is very dear to my heart. It has brought a lot of benefit to agriculture and trade. It has been used since the 50s about, and the IAEA, in particular, the laboratory Cyberstorf, is really extremely competent in this field and has trained many people around the world. So the idea is to manage a population of insects that are detrimental to agriculture, to crop production. We want to reduce their population, and sometimes, but that's more difficult to do, to eradicate this insect. This has been achieved actually in favorable context where the area is limited. For example, on a small island, it's easier to do than in the middle of a continent with the agriculture all over the place, and insects able to come easily from a neighboring area. So the technique is based on sterilization, so the inability effects on the spermagonia, but on the males. So the technique is based on the mass rearing of insects, and these insects will be irradiated, and after the males, only the males are taken and will be dropped using a plane or more and more a drone on an area that is infested with this insect. When a sterile male will meet a wild female, there will be no offsprings. And that's how the population of these insects will progressively decrease. So this is a very simple explanation, and invite you to see the literature of IAEA, which is, it's extraordinary. There are many examples, but I want to show this one that was given by Vanessa Diaz during the IMRP meeting in Bangkok. The New World screwworm was a pest that had terrible effects, especially in the Americas. It's a horrible bug because it eats you alive. You see the disgusting picture. It will eat your flesh. So it was a huge problem, and the sterile insect technique has been used to combat it. And it has been estimated that for a cost of 1, the benefit would be 33. So hugely beneficial application of irradiation that is not very well known because it does not happen. It's not very visible. It does not happen in industry. It's not a commercial application, but however, a very useful application. Irradiation as a phytosanitary treatment is something rather new. that started really less than 20 years ago with in Hawaii sending irradiated fruit and vegetables, in particular sweet potatoes, to the continental US. These had been treated because Hawaii has insects that the US do not have and do not want. So there are different ways to apply what is called a quarantine measure or phytosanitary treatment to fruit and vegetables. One of the most common is to fumigate them with methyl bromide, an ozone depleting gas, or using cold treatment or hot water deeping. In the case of irradiation, all fruit and vegetable can be treated and only one dose can be used for a specific insect. So the interesting thing is that the purpose of irradiation is not necessarily killing the insect. It's not necessarily mortality. The insects are a bit complicated. Here you have the cycle of a fruit fly, one of the most damaging insects. And so they start at eggs and then they become a caterpillar, then they become a pupa. Well, there are different names, and again, we're not going to go deep into entomology, but irradiation can block the evolution from one stage to the other. So, for example, it can make it the eggs, if they have been irradiated, will not hatch. So you will not go to the second stage of being a caterpillar, or the pupa will not become an adult. So different types of effects, again, due to different types of damage on the insects, depending on the stage where we are, and of course depending on the dose. But you want the dose to be as low as possible, because the lower the dose, the less effect you will have on the product itself. But you must be sure that the minimum dose must be there to achieve the wanted effect. So the fact that a product has been properly treated, but there are still live insects in it, is really a change of paradigm and a shock to many biosecurity inspectors, quarantine inspectors, because it's not, their background is, a product is okay if there is no live insect in it. And it's not the case with irradiation. So you can have live insects, but they will be sterile. And 20 years of experience have seen that this works indeed. And the confidence in the treatment is based, well, first on research, determine properly the dose that it needed to achieve the effect. So for example, no emergence of adults. The fact that the dosimetry has been correctly done. And third, that precautions have been taken to prevent infestation before treatment or reinfestation after irradiation treatment of the products. And there is an interesting growth of this application, in particular in America. US and first importing country for irradiated produce, and also in Australasia, in particular Australia. Finally, we're going to conclude with pasteurization and sterilization. So definition, simple definition, because depending on what sector you work in, you will see slightly different definition. But I think this one can reconcile most definition. Pasteurization, the purpose is generally to render the product safe. Not always, because it can also, the aim can also be to extend shelf life. So you're going to eliminate whatever eliminates means, the undesirable microorganisms, not necessarily all, as opposed to sterilization, where you want to kill them all. So, of course, we all know pasteurized milk or sterilized milk, and this is the reference method. So radiation is another way to pasteurize or sterilize. And interesting, because there will be no temperature increase as opposed to heat. It can be used for frozen products. You can pasteurize a frozen product, which is not possible with heat pasteurization, or you can pasteurize products that have the form of a powder, and heating is very difficult to apply. Like other sterilization or pasteurization method, it can be done through the final package. And the beauty of irradiation is that you have only one control parameter, which is dose. So eliminate, kill, destroy, etc. I prefer the word inactivation. Inactivation being the transformation that will make an organism unable to multiply, to reproduce itself, and therefore to produce colony forming units. Because how do we know that an organism is not alive? Is by trying to make it alive by growing it, for example, on an appropriate medium. So inactivation by irradiation, it's an exponential law. So in theory, you will never get to zero. A concept that is commonly used is the D10, the dose by which you will reduce by 1/10 the contamination, so the number of colony formed units. So this D10 will depend, of course, varies with microorganisms, but can also be influenced by temperature, by presence or absence of oxygen, by humidity. Remember, humidity, water, indirect route, so more efficient, and the substrate, because in a substrate, let's say in the matter that carries the microorganisms, you can have substances, compounds that will act as a protection against irradiation, or by, for example, scavenging the radicals, or that can make them more sensitive to irradiation. So radiation pasteurization can be used and is used for food. I put this word on the top right, radicidation, that has been mostly forgotten, and that was put forward by Americans in the 60s, and I like this word actually. So when you want to inactivate Salmonella, Listeria, etc., all these bugs that cause food poisoning, that's radicidation. And we're lucky because many of these pathogen organisms, foodborne pathogens, are rather sensitive to irradiation. So rather low doses will be enough to inactivate them, meaning doses that will have minimum effects on undesirable effects on the food. Unfortunately, I think this application has a huge potential, but it's not very widely used. So we can give a few examples, but it's on a limited scale. Ground beef, irradiated ground beef is sold in the United States, where there was a scare after outbreaks of E. coli. In France and Belgium and Netherlands, you can find frozen frog legs in supermarkets that were irradiated. And in the 7-Eleven stores in Thailand, you will find fermented sausages that have been irradiated because they can cause very serious food poisoning if they are not irradiated. Radiation pasteurization of food in the sense that was given by the word radorization. So you're not aiming at a pathogen, but you want to reduce the microbial load of a product, especially when it is an ingredient. So we know that spices, they are natural products. So as natural products, they are naturally contaminated by a number of microorganisms. And good manufacturing practice will require you to be very clean in your process, etc. So you cannot use dirty ingredients and contaminate the whole vat of sausage meat that you are preparing, for example. So the dose, this has been applied mostly to dry food ingredients, so no water, so direct route, more difficult to inactivate. And you have here an example. You see on the list of ingredients of this steak sauce found on in South Africa that the spices have been irradiated, and this is the main application of irradiation around the world. Now, I'm going to finish with the second most important application of radiation processing, very widely used, for which there is a great harmonization worldwide based on the ISO 11137 standards that are made of several standards, actually. We know that for the safety of patients, some medical devices, those that are implanted, for example, those that surgeon gloves, for example, that will come in contact with the bloodstream must be sterile. But in spite of all the precautions taken by manufacturers, clean rooms, etc., there is always a bit of contamination, and also contamination once the product inside the package, there will be a contamination, hence the need to sterilize them. So what does it mean, sterile instrument? Well, it's an instrument where there is no viable organism, microorganism. How do you know that there is no viable microorganism when you have manufactured, let's say, 10,000 syringes in one day? You would have to put, if you wanted to be sure, you would have to destroy them all and sterility test them, which of course is not possible. So until the 80s, 90s, in many countries, they were satisfied with taking, say, 50 samples out of this 100,000 and checking if they were sterile or not. And if they were sterile, they were happy. The whole lot was considered sterile. This has no actually statistic significance. And we got away from this. And the concept that is now used is the sterility assurance level, which is a probability of a viable organism being present on a product unit after sterilization. And this is actually a more practical method. And this sterility assurance level will be expressed as 10 to the minus n to power minus n, meaning the probability of finding one viable microorganism in 10 power n. The common value is 10 to the power 6, so 1 in a million items manufactured and sterilized. So here you have the curve giving in a log scale, you know, after you start destroying the organisms that are present on the medical device. As you increase the dose, you increase the probability of not finding one on a given number of instruments. So methods have been developed from the 90s and now well known and widely used to establish the dose and periodically check that this dose is still good for the sterility assurance level that is designed. So there we're talking about destroying the whole population and inactivation of microorganisms. And with this, I finish my presentation, and I thank you very much for your attention.